Well, now that we've gone from atoms up to the building blocks of life, which we learned were amino acids and the letters of DNA and RNA, now we have to take those building blocks and start building the next items. So today we're going to look at how amino acids build proteins. As we said last time, the amino acids are allowed to link together because of their amino and acid ends, and so they form long chains. These chains are what we call proteins. So they do 90% of the jobs in the cell because they fold up into machines. You probably have heard about enzymes. Enzymes are protein machines that catalyze, which means to speed up chemical reactions. And technically, enzymes are a form of molecular machine. But when most people say molecular machines, they think of proteins that literally push or pull or walk or spin or do some other form of force or motion. So earlier in this series, we talked about a molecular machine that I study that's called kinesin. And that's a molecular machine that literally walks one step in front of the other along strands of proteins inside of the cell. So that would definitely be a molecular machine. Molecular machines usually catalyze ATP breakdown. So in that sense, they also are enzymes that are catalyzing a reaction. The reaction is to break down ATP, which, as we learned about before, was adenosine triphosphate, and it's the currency of energy. So it's just burning the energy inside the cell. That's an enzyme job, and that's what molecular machines have to do. Well, I said that around 90% of the jobs in the cell are done by proteins. It's just kind of a rough number. The other number of jobs are done by RNA machines or combinations of RNA protein machines. For example, the ribosome, which is the machine that translates RNA into protein. You may remember that translation is a word that now means moving from the RNA language of 4 letters to the protein language of 20 or 21 letters. That ribosome, that machine, is made up mostly of RNA, and especially in the parts that do the actual job of putting things together, that part is RNA. And ribozymes, which is kind of a name that has been coined with RNA, ribonucleic acid, and enzymes, put together, ribozymes are RNA enzymes. And some of them don't even need proteins around to do their jobs. But for the most part, proteins are what do the jobs inside of the cell. And they only do it after they fold up into a specific shape. So if you have a long and skinny train of amino acids all stuck together nicely in the right order, that still won't do the job. You need to let go of the ends. And with the hydrophobic effect that we talked about earlier, the parts that are greasy will fold up into the middle. They'll try to get away from the water. They're afraid of water. They'll go into the center, and it will perfectly line up the order of all the amino acids into just the right shape to perform a job. Now, how it does that, going from the sequence of which building block comes after which building block, is a completely determined event. If you do it 1,000 times, it'll always do it the same way, as long as it's a pretty simple protein. But we don't know how it does it. We can't even model this on our fastest computers. So we're still trying to figure that out. Biology is still making progress in understanding this mystery of how it goes from the primary sequence into a folded up shape. Proteins are made of roughly 100 of these building blocks, so that means roughly 1,000 atoms. And that's because they are kind of like Legos. They need to have a big enough shape in order to do their job. Just remember the Lego pliers that you had to mentally make in your mind. They have to have a large number of Legos in order to do that job. Similarly, proteins have to have a large number of building blocks. Now, an activity to help understand what exactly is happening when protein folds up into its shape is to think about salad dressing. Okay? Now, you've all taken salad dressing and shaken it up and watched all the little particles of oil mixed with particles of water, and then it settles out so that the oil floats to the top and the water goes to the bottom. You might think that that's happening purely because of gravity, and that if you took this salad dressing out in space in zero gravity, maybe they would just mix together perfectly. But think again, because that is not what would happen. Oil does not like water. Even if the oil was the exact same density and weight as the water, they still would want to separate from one another. Okay? Now, another thought experiment to do, and besides just thinking about salad dressing and zero G, is to think about a necklace of water molecules that were decorated with little pieces of oil. Okay? So they have this necklace of little water droplets and little drops of oil hanging underneath them, all on a nice little silk cord. What would happen when you let go of it? Maybe in space with no gravity. Again, the water molecules and the oil molecules want to try to get away from each other. So the water molecules and their little chain or their little string would go around the oil. And that's exactly what happens in these proteins as they fold up. It's also how DNA gets its double helical shape too, because the inside, those letters, those rings, are what are kind of oily and greasy, and the outside, the phosphorus, or some people thought it was going to be arsenic, and the other parts of it are very watery, so they go to the outside. Now, as a final sort of story, we're going to look at now how some people saw the code that takes the 4 letters of RNA and maps them to the 20 letters of protein as a frozen accident. So somewhere in the history of the Earth, a certain code was created that said these 3 letters of RNA will always map to this 1 letter of protein, the amino acid building blocks of protein. But it turns out that if that were true, then if we scrambled up the code that we have and made a new code, one that doesn't exist in nature, then there should be a 50-50 chance that the new code we make is better than the old code. And by better, I mean better at preventing dangerous mutations from happening inside of an organism. Okay? Well, it turns out that's not true. In 1998, a scientist named Stephen Freeland and another scientist, Lauren Hurst, looked at the codon table, generated some fake codons, and they found that unless they did 1 million different new codons, they couldn't find a single a million different codes. They couldn't find a single code that was better than the one that we have here on planet Earth. So they called it the 1 in a million code. And subsequent experiments showed that it might not only be 1 in a million, it might be the best of all possible codes. That's how they subtitled one of their later papers. And it is because the arrangement of which amino acids are encoded by which 3 letters is not random at all. Similar amino acids get mapped to similar codes, 3-letter codes, and that keeps everything more organized. So if you can imagine, again, this necklace of watery droplets and oily droplets. And if you change, let's say, the size of one of the watery drops or the oily drops by a lot, it might not fold up into the same shape. But if you could keep them roughly the same size, then even if it changed a little bit, it wouldn't mess up the entire fold. And that's what happens thanks to our best of all possible codons tables. Well, if you're doing a little more extra reading, I would suggest that you read the leaf, What Are Amino Acids again, which is a little review, but it will help you. And also review How Do Cells Read and Execute the Instructions in DNA, that branch and all of its leaves. Also, you can now go ahead and read Why Do Cells Need So Many Proteins? This is a good time to learn about how proteins do all of the jobs inside of the cell. Also, try to picture how the combination of hydrophobic and non-hydrophobic amino acids work to build proteins, and look at the specifics of how ribosomes assemble proteins so that you can understand when I talk about these 3 letters going to 1 letters and makes a little more sense to you.